In this video, I'm going to walk you through Lab 9. Lab 9 takes a look at the different morphologies of the eukaryotic microorganisms that we will study throughout the semester. This first image that you see here is of a fungus. In particular, this is Penicillium. And what you see here are its reproductive structures. This long structure here that you see is called a conidiophore. And at the end of the conidiophore, you have its reproductive structures, the conidiospores. So each one of these is a little individual structure, and then these will be lifted off by the wind or birds or other organisms and then drift around until they find a place to land. The conidiophores and conidiospores are microscopic parts of the mold, and they exist on the kind of furry portion of the mold. Each one of those little hair-like structures of a mold are called hyphae for plural, or hypha singular, and a bunch of those, a group of those, like when we see a mold growing on a fruit or vegetable, the group of those is called a mycelium. So what we're seeing here with these conidiospores of Penicillium is that these structures would exist on the end of the aerial hyphae. So they'd be kind of sticking up in the air to be able to be distributed wherever. Now, conidiospores are going to be different from our next reproductive structure that we're going to see, the sporangiospores. These are the sporangiospores of a mold called Rhizopus. Now, we still have hyphae that we can see in the background, but instead of the spores just being loose, we're going to have this structure called a sporangium that's going to be filled with the sporangiospores. So all of these little dots that you see within here. On this particular sporangium here, we can see that it's about to rupture, and so all of these spores then would be released into the atmosphere. So they're not just sitting there loose like they are on the Penicillium mold that we just saw. They are actually contained within a sac. The third type of mold spores or reproductive structures that we'll see are arthrospores. These are arthrospores, or sometimes referred to as arthroconidia, of a particular fungus called Coccidioides. Now you'll notice these look completely different because these spores, or these reproductive structures, are actually pieces of the hyphae that are going to break off or fragment. So what you're seeing here would be a hypha. Okay, and you see that it's fragmenting and pieces are breaking off of it. So it doesn't necessarily have the spores that we saw with Penicillium and Rhizopus. The hypha itself is just going to fragment, and each one of those pieces is going to serve as a reproductive structure. The next image that we have here is of Candida albicans. Now, Candida albicans is a fungus also, but it is considered a yeast. Yeast are single-celled organisms. So what you're seeing here, these round structures here, this would be the cell of Candida albicans. Candida is considered dimorphic, meaning that they can exist in multiple ways or multiple configurations. What we see here, if we just had this little blue portion here, that would be considered a typical budding yeast. But instead of just acting like a typical yeast, by being dimorphic, Candida albicans can actually develop a hypha like a mold would. And these hypha can be used to actually allow the candida to burrow deeper into tissues once it, is causing an infection. So it can go from this yeast form or this single-celled, small circular cellular structure to this filamentous form because it's dimorphic. It can exist in two different structures. Switching gears from our fungi, now we take a look at some of our algae. So in this image here, there are actually 3 different algae that you're going to draw, but we're going to hold off on the diatom. Right now, we're just going to focus on the Euglena and the Volvox. So Euglena is number 5, and Volvox is number 8. So here we have Euglena, and you'll notice that it's green because of its chlorophyll. Euglena, like other algae, is photosynthetic, so it will be able to absorb sunlight and make its own food. You'll see this little area here that looks like it's almost a hole in the cell, and this can be used to take in water and then eject that water. That kind of helps Euglena move throughout its environment because it will live in an aquatic environment. And so Euglena serve as algae as food for typically freshwater organisms. We'll find this typically in freshwater. Now, when we look at Volvox, we'll see something a little bit different. With Volvox, you kind of see these larger circular structures with all these little smaller structures in them. Volvox, also an algae, you can see that it's green, also has chlorophyll, is photosynthetic, but Volvox algae can live communally to where they look like, these kind of look like one organism with multiple different cells, but really this is a community of algae that live together. Volvox kind of takes on almost a multicellular condition. They kind of bridge the gap really between what we would see with the algae, a typical algae, and a plant. So they're, in terms of their sophistication or their complexity, they're more complex than something like Euglena or Spirogyra that we see here. The next alga that we have here is Oscillatoria. This is number 6. And Oscillatoria are just these long green, again, green structures here that almost look like blades of grass. They're bright green in color. These would also be or live in fresh water. And they also perform photosynthesis. So these would be like Euglena. They would serve, as food for other organisms that live in those freshwater environments. So this is Oscillatoria. Another alga that we'll look at here, this is Anabina. This is number 7 in your lab. Anabina almost looks like a strand of green pearls. You see all of these little cells that are kind of all attached together. And again, they would live in fresh water typically and serve, at the bottom rung of the food chain, serve as food for, fish and other organisms that live in the water. So this is Anabina. This wide array of cells that you see before you are the Dinoflagellates. This is number 9. Dinoflagellates are typically considered to be algae, but in some cases they're considered to be protozoans. It really depends on what source you look at, what source you read, how these are classified. Dinoflagellates can photosynthesize. Some of them can photosynthesize, and then some of them are heterotrophs that actually eat other dinoflagellates or eat the algae that we were just looking at. So they have a wide variety of sizes and shapes. Some dinoflagellates can produce toxins that can be deadly to fish that live in the water. So when we look at these different dinoflagellates, they all have a different role in, food webs and food chains in both freshwater and marine environments. So dinoflagellates we can see in freshwater lakes and ponds, but we can also see certain types of dinoflagellates in the ocean. So these are diatoms, number 10 in your lab. Diatoms are considered to be algae, but they are completely different from all of the other forms of algae that we have looked at. Diatoms have cell walls that contain silica, and silica is the base ingredient or substance that makes up glass. So when you look at all of these diatoms, they look shiny, like they're reflecting light, and that's because of the silica that we find in their cell walls. Just like the dinoflagellates, they're going to come in all shapes and sizes. I love looking at different pictures of diatoms. It reminds me of looking through a kaleidoscope when I was a kid, because of the different shapes and sizes and even different colors as they reflect different wavelengths of light. Diatoms are typically found in the ocean, although we can find them in freshwater as well. Switching gears again now to protozoans. Remember, protozoans are closely related to algae in terms of their kingdom that they're in. They're both in the protist kingdom, but the protozoans have more in common with animals than they do plants, in that they don't photosynthesize, and a lot of times they don't have a cell wall. So what we see in this image, these structures that you see in the background here, these are red blood cells. So what we're looking at here is a blood smear, and we are seeing two different forms of Plasmodium. We have the gametocyte form over here of Plasmodium falciparum, and then over here within this particular red blood cell, we have a growing trophozoite of Plasmodium malariae, which are two of the causative agents of malaria. Plasmodium has a very complicated life cycle. We'll get into that later on in the semester. So here we have another protozoan. This is Giardia lamblia. This is number 12 for Lab 9. I like looking at pictures of Giardia because it looks like he has a face and with eyes and a nose and a mouth. But in reality, what we have here is a single-celled protozoan. These little spots here that look like eyes are actually sucking discs that allow the Giardia cells to attach to the walls of the intestine. You'll notice these little wispy structures here. Those are its flagella that allow it to swim through its environment, which is going to be freshwater lakes and streams. Switching gears one last time to the helminths, our parasitic worms. And this is not exactly a worm, but it is classified in with the parasitic worms. This is a liver fluke. This is Fasciola hepatica, and it is a parasite that infects the liver. We'll talk about its complicated life cycle when we discuss our gastrointestinal diseases. This is the mouth parts of Tienia pisiformis, which is a tapeworm. And what you see here, these circular structures are sucking discs, and these structures up here that kind of look like eyelashes are actually hooks that they'll use to attach to the intestinal wall. So this is Tienia pisiformis, the tapeworm. Our last parasitic worm here is Trichinella spiralis, which is a roundworm.